Hello students, welcome to EPG Pathshala. I am Vidya-mitra, former professor of Biophysics at All India Institute of Medical Sciences, Delhi, at JP Institute of Information Technology, Noida, and former professor of Bioinformatics and director at Dr. D.Y. Patil Biotechnology and Bioinformatics Institute, Pune. Today, I am going to discuss about a module, interactions of small molecules with DNA. This comes under paper Biomolecules and their interaction. Small molecules that bind to genomic DNA can serve as effective anti-cancer, antibiotic, antiviral therapeutic agents. With the completion of Human Genome Project and developments in the experimental medicine, biochemistry, and biotechnology, understanding interactions between small molecules and DNA would be of great importance. It can give a quantum gem in the development of DNA binding pharmaceuticals. DNA molecule has two primary functions: replication and transcription. Many small molecules can bind to different regions of replicating fork of genomic DNA and interfere with the function of DNA polymerase, which controls replication of DNA. Several metabolites, drugs, and carcinogens can enter the cell, then bind to specific cytosolic receptors, and then the complex together controls transcription of specific segment of DNA and synthesis of proteins thereby. Let me first explain you what we are going to learn in this module. First, I want to clarify you the basic concepts in small molecule DNA interactions. Then, I want to give you some details about the experimental and theoretical methods used for the study. This is very important, how we are going to study these interactions. Next, I want to describe different ways how small molecules can bind to DNA. These are external binding mode, intercalation, minor groove binding mode, major groove binding mode, and mixed binding mode. Then we'll do binding by alkylation, then binding at the three-way junction. Lastly, I want to introduce you to one of the most important area, that is designing of a small molecule which can specifically interact with DNA and have a specific influence on DNA. Basic concepts in small molecules DNA interaction. Small molecules interact with DNA by non-bonded, electrostatic, and H-bonding interactions with bases and backbone. Water, alkali, and alkaline earth metal ions bind to sugar phosphate backbone externally. Transition metal ions such as copper, zinc, and platinum bind with DNA bases. Aromatic and heteroaromatic molecules can slide in between the base pairs and intercalate. These are stacking and electrostatic interaction with bases. Heterocyclic aromatic molecules with cationic tails and protruding NH or H groups can interact with DNA by H bonding, hydrophobic and electrostatic interactions in the DNA minor and major growths and recognize DNA base sequence, whereas aromatic and heteroaromatic compounds with protruding ring structures can have a mixed mode binding. Variety of experimental and theoretical techniques are used for study of small molecule DNA binding. For example, X-ray fiber diffraction, UV, VIS, IR, CD, fluorescence spectroscopy, linear dichroism can be used. Sedimentation coefficient, differential thermal analysis, and electron microscopy has been used. Nuclear magnetic resonance spectroscopy has been used. Single crystal X-ray diffraction has been used. Variety of theoretical techniques as empirical, semi-empirical, quantum chemical, molecular mechanics, molecular dynamics, free energy calculation, ab initio calculations have been used. Similarly, DNA footprinting technique has been used to study small molecule DNA interaction. Ligand DNA complex structures. Many structures are now stored at nucleic acid database at Rutgers University. This can be easily available at the server ndbserver.rutgers.edu. They have only DNA structures 1600, drug DNA structures 366, peptide DNA structures 13, protein DNA 3462. The data as on 1st September 2015. Many structures are also available at Protein Data Bank PDB, at RCSB Research Collaboratory for Structural Bioinformatics. Again, site is http://rcsb.org. External binding. Few small molecules, water and ions, as magnesium 2+, calcium 2+, and sodium plus can attach to the outer surface sugar phosphate backbone, mostly through salt linkage, electrostatic, and H bonding interactions, leading to a structural and conformational changes in DNA. Although these interactions are of paramount importance in biochemistry, these are usually non-specific. Let me explain you what is intercalation. Certain flat, aromatic, and heteroaromatic molecules, this can slide in between the base pairs and stabilize the duplex without disturbing base pairing. This leads to lengthening of the DNA duplex around 3 angstrom per bound drug molecule. This also causes unwinding of the DNA by 5 to 26 degrees. Intercalation prevents replication by interfering with the action of topoisomerases. The degree of unwinding depends on the structure of intercalating molecule and the site of its intercalation. We show in this picture three intercalators: a masaccharine, proflavine, and Ethidium Bromide. There are many more structures of intercalators with DNA are available in databases. There are about 200 structures now available, and one can study how these drugs bind to DNA. There is a classical or an end-on intercalation model. This model was given by Lerman in 1971, and this is shown in this picture. That is, left-hand side picture is of normal DNA and the base pair shown stacked over each other. On the right-hand side, it is a distorted DNA, but the black here is a intercalator which fits into the base pair, and the backbone gets deformed because of unwinding of DNA. In this particular case, the long axis of DNA, C6-pyrimidine on C8-pyrimidine, is usually parallel to the long axis in the drug, and the dimensions of the ligand are usually similar or close to the DNA base pair geometry. There is a classical or an end-on intercalation model. This model was given by Lerman in 1971, and this is shown in this picture. That is, left-hand side picture is of normal DNA and the base pair shown stacked over each other. On the right-hand side, it is a distorted DNA, but the black here is a intercalator which fits into the base pair, and the backbone gets deformed because of unwinding of DNA. In this particular case, the long axis of DNA, C6-pyrimidine on C8-pyrimidine, is usually parallel to the long axis in the drug, and the dimensions of the ligand are usually similar or close to the DNA base pair geometry. We show in this picture the structure of Ethidium Bromide with a dinucleotide sequence ApU Duplex. Only few atoms of the ligand protrude out of the planar structure, as in the case of Ethidium Bromide. The ligand rotates around its own axis so that these groups can protrude in the major or minor grooves. The structure shows Ethidium Bromide with dinucleotide duplex ApU. The Cris-Cross Model. In the Cris-Cross Model, which is shown in this picture on the right-hand side, the long axis of DNA is rotated with respect to the long axis of the ligand. The ligand long axis is more closer to the diad axis of DNA. This is seen in the case of Daunomycin with the hexanucleotide duplex CGTACG. This was very important because this was the first high-resolution ligand DNA structure at 1.5 angstrom resolution. We see here Daunomycin in the base pair 8TA in the hexanucleotide duplex. The sugar pucker and glycosyl conformation. Crystal structures of now more than 200 intercalators with DNA are solved. In case of Ethidium Bromide, proflavine, acridine orange with dinucleotide monophosphate duplex, it was observed that purines at 5' end had C3' endosugar pucker, while pyrimidines at 3' end was with C2' endoconformation. The glycosyl torsion angle in most cases was anti-conformation. Brew recognition. Many small molecules like Netropsin, Distamycin, Hoechst 258 bind to DNA in the minor groove of 80-rich sequences. These molecules recognize DNA base sequence and do not alter DNA base pairing or its length. The question now arises is how these molecules recognize DNA base sequence, because only regions where bases are visible to small molecules are the floor of minor or major grooves. The edge bonding donors and acceptors in the major and minor grooves of GC, AT, CG and TA base pairs are different. We show in this picture the minor groove side and the major groove side of these four sequences. Here W1, W2, W2', W1' show major groove functional groups which can interact with the ligand, and S1, S2, S1' show the minor groove binding specific groups. And you can immediately notice the differences between these groups in these four base pairs GC, AT, CG and TA. The recognition is through H bonding with donors or acceptors in the minor or major group. The methyl group of thymine and corresponding hydrogens of cytosine provide additional discrimination. We show in this picture a schematic representation of electron donor acceptor pattern in DNA grooves. Here in this picture, the red is an acceptor which is an oxygen atom. Blue is nitrogen which is also an acceptor. Black is NH, electron donor. Green is also electron donor, but it is CH donor. We show here the sequence GC, major groove, minor groove. AT, major, minor. CG, major, minor. TA, major and minor. And from the color difference, you can immediately make out there is a pattern, and the ligand recognize this pattern. Minor groove recognition. There are now over 110 structures available of ligands binding in minor groove of DNA. Netropsin and Distamycin are polyamides with antibiotic and antiviral activity. Netropsin is active against gram-positive and gram-negative bacteria. Distamycin shows anti-cancer activity. These molecules belong to the class of Lexitropsines. These have pyrrole rings connected by peptide bonds. These also have cationic tails. This is shown in this slide. The structure of Distamycin and Netropsin. The two adjacent pyrrole rings are oriented with respect to each other and at an angle ranging from 18 degrees to 20 degrees. These have semicircular shape like a necklace. Netropsin and Distamycin. The width and curvature of these molecules is suitable for binding in minor groove of DNA. Steric compatibility was found to be an important factor for their interaction with DNA. These interact with 80-rich region of DNA. Interaction is by H bonding and hydrophobic forces. The terminal amidine group is basic and serves to attract the drug molecule to the negatively charged phosphodiester backbone. The backbone could be reduced by incorporation of urea, which shows the electrostatic interaction is important. We show two beautiful pictures of interaction of netropsin and distamycin in minor groove of DNA. Both the structures are obtained with the sequence CGCGAATTCGCG, and the first structure is by Kopkaita PDBID 101D, second structure is by Call et al. By PDBID 2D and D. The structures are quite similar to each other. Let's see more details about the interaction of netropsin with dodecamer CGCGAATTCGCG. Netropsin binds in minor groove, displacing the water molecule in the spine of hydration. Netropsin amide NH furnish hydrogen bonds to bridge adenine N3 and thymine O2 atoms occurring on adjacent base pairs of opposite helix strands. It sits symmetrically in the center with its two parallel rings slightly non-planar, so that each ring is parallel to the wall of its respective regions of the groove. Netropsin DNA interaction. The drug binding neither unwinds nor elongates the double helix, but it does opens the minor groove by 0.5 to 2 angstrom. It bends back the helix axis by 8 degrees because the normal structure of dodecamer was bent by 90 degrees. By binding of netropsin, the bending reduces. So, it reduces in the region of attachment. The netropsin molecule has an intrinsic twist which favors insertion in minor groove of BDNA. It is given a small additional twist upon binding. The base specificity is provided not by hydrogen bonding, but by loose van der Waals contacts between adenine C2 and adenine C2 hydrogens and CH groups of pyrrole rings of the drug molecule. Let me now explain you the interaction between distamycin with DNA. Distamycin can bind DNA exactly in a very similar way as netropsin in the minor groove of AT-rich sequences by forming H bonds and also having hydrophobic interactions. In case of GC-rich sequences, the 2-amino group of guanine prevents distamycin from binding in the minor groove because of steric hindrance, thus conferring AT selectivity on the drug molecule. Binding of Distamycin A and SG2057-DNA in 2:1 complex. 2-Distamycin A or SG2057 molecules are bound side by side with a dyad symmetry in an antiparallel orientation in expanded minor groove of DNA. This is one of the most interesting structure and first seen by Sundarlingam in case of Distamycin A and by now for number of other Lexitropsines. Distamycin A with a DNA sequence ICICICIC. The The structure was given by Sundarlingam. In this case, the amides of each drug molecule are hydrogen bonded to the minor group base atoms of one DNA strand. DNA duplex also exhibits striking low-high alterations in helical twist angles, sugar puckering, and the phosphate conformations, providing the basis for a new model for an alternating bDNA with a dinucleotide repeat. Normally, all bDNA structures are with the mononucleotide repeats. Only zDNA has a dinucleotide repeat. So, this structure, the repeat was dinucleotide instead of mononucleotide. We show in this picture fluorescent dyes Hoechst 33258 bind also to AT-rich region of CGCGAATTCGCG, similar to netropsin and distamycin. The structure has been explored both by linear dichroism and single crystal diffraction techniques. Lexitropsines. Lexitropsines are semi-synthetic ligands. These are shown in the picture here. There are various possible side groups which can be attached, like CH, N, and so on. Series of dimers and trimers of distamycin, netropsin, imidazole rings have been synthesized and studied in order to increase DNA binding region to 10 base pairs or more in an effort to design small molecules which can recognize any DNA base sequence. This is a art of designing, how you can design a specific ligand to recognize any sequence of DNA. Lexitropsin were seen to form complex with DNA in stoichiometry, both 1:1 and 2:1 in minor group at sequence 5-AATTC with its bulky piperazine group extending over the CG base pair. The metahydroxyl group was found in two distinct orientations, neither of which participates in any direct hydrogen bonds to the exocyclic amino group of the guanine base. Major group recognition. There are not many small molecules which bind to DNA in the major group. The total number of available structures is very small, close to 10. Methylgreen is a DNA major binding drug. The interaction with poly-DA DT, poly-DA poly-DT, and triplex poly-DA 2 poly-DT was studied with linear dichroism. A typical example of major group binding is a third DNA strand with a complementary edge bonding groups which can wrap around DNA in the major group and make a triple helix. Mixed mode of binding. Several studies on bifunctional peptide antibiotics, Echinomycin and tandem, using X-ray diffraction and theoretical modeling showed that these also do not disrupt Watson-Crick base pairing. These bind in the minor group with partial intercalation, and this serve as a very good model for protein-DNA interaction. We show in this picture bis intercalation depsipeptide, Echinomycin and triostin binding to DNA. Both of these bind partially in the minor group and partially with the intercalation. So they all have a mixed mode of binding. On the left hand side, we show structure of Echinomycin, PDB-ID 3GO3. On the right hand side, we show the structure of triostin with GATATC. The PDB-ID of this structure is 185D. You can use any graphic program and explore the structure yourself. One of the most interesting drug DNA binding structure study has been Daunomycin with CGTACG. Why it is so important is all the structures of intercalators with DNA were with dinucleotide duplexes, and this was the first example where more than half a turn of DNA hexanucleotide, that is CGTACG, was used. Another important thing was the structure was solved at a very high resolution, 1.5 angstrom, and it was done by Nobel laureate Alexander Rich. We show first the nomenclature for Daunomycin. There are 4 rings, A, B, C, D, and the A ring is attached to the sugar group. The next picture shows Daunomycin DNA structure. We explain it in the next slide. So the PDB-ID of this structure is 1D11. The Daunomycin intercalates in Cris-Cross model. We have talked about it earlier. The D ring of the drug is protruding in major group, whereas A ring with the sugar group occupies the minor group. The O9 of Daunomycin H bonds with NH2 group of guanine, and O13 of Daunomycin H bonds with O2 of cytosine. Sugar pucker for all the 6 bases belongs to C2-endo family, not C2-endo, C3-endo as in case of intercalators with dinucleotide duplexes. The 6 bases in this case are sugar puckers: C2-endo, C1-exo, O4-endo, C2-endo, C2-endo, and C3-exo. Most important point is the Daunomycin does not recognize any specific base sequence, as N3 of guanine and O2 of cytosine both are non-specific. DNA binding by alkylation. This is seen for cis-platin complexes. Metal atoms as platinum form H bonds directly with DNA bases and bind by alkylation. They have been in clinical use from a very long time for the treatment of cancer. The typical examples are carboplatin, nedaplatin, oxaliplatin, and they have been extensively used for cancer treatment. We show in this picture the structure of how platinum binds to the DNA molecule, and this is shown as the cis-platin with chlorine and 2 amino group rather, and this interaction almost stops DNA replication and kills the cell. A new mode of binding has been very recently discovered by Mike Hannon and McWell-Call. This had been very interesting because they found a ligand which has a very perfect, beyond your imagination, fit into the DNA in the 3-way junction. This is important because 3-way junctions are found in many disease conditions as Huntington's disease, myotonic dystrophy, during cancer growth, etc. And it will give a new avenue for designing of the drugs against cancer. One of the most interesting way how a small molecule binds to DNA is binding of a ligand in a three-way junction. The authors have shown that a cylinder of 6 phenyl rings form almost a perfect pi stacking interaction with 6 DNA bases that are placed at a three-way junction of 3 double-stranded arms that come together. There are several non-covalent forces that hold the drug in this three-way junction. Face-to-face stacking. In the three-way junction binding, another interesting thing which was noted by the authors was the planar aromatic rings have perfect stacking with each other and they stack on the DNA basis. And this technique has been used for designing molecules with non-covalent interactions with DNA. The design strategy is based on understanding the mechanism of recognition of DNA-based sequence by small molecules. The first time when designing was attempted, that was by Zimmer's group for methylpyrrole groups. He showed that if you change methylpyrrole groups, it increases the stability of the binding. Next, isohelical analysis was given by Dickerson's group. He suggested that long flexible molecules with proper curvatures can bind with DNA and recognize DNA. So one can design such molecules, long flexible molecules with proper curvatures. One of the most interesting designing strategy was given by Derwan's group. These were called isolexins. Derwan took this approach further in synthesizing isolexin, a series of oligomeric hairpin polyamide molecules containing pyrrole and imidazole ring system that are able to bind side by side in the minor groove of DNA with a very high affinity and in a sequence-specific manner. So students, what we have learned in this presentation. First, we tried to understand the basic concepts of small molecule DNA interactions. Next, we have gone over through the different experimental and theoretical methods used for study of ligand DNA interaction. Third, small molecules DNA binding and recognition by intercalation, minor groove and major groove binding, mixed mode binding, alkylation, and binding in a three-way junction is explained to you. We have cited specific examples in these cases. For example, for intercalation, we gave example of Ethidium Bromide. For minor groove binding, we gave example of Netropsin, Distamycin, Hoechst 33258, Lexitropsines. For mixed mode binding, we gave examples of Echinomycin and Daunomycin. Alkylation example is cis-platin compounds and new synthetic molecules which bind to three-way junction are also explained to you. I am really thankful to you for your kind attention and explaining you the importance of small molecule or ligands with DNA interaction. Thank you.